Hello everyone, and welcome back to another GCSE Biology revision session. Today we're exploring light and electron microscopes, essential knowledge for your exams. This video is part of the Microscopes and Magnification series, so make sure to check out the other videos in this playlist. Let's get started. By the end of this video, you'll be able to describe the features of light and electron microscopes and compare the features of light and electron microscopes. Let's start by looking at light microscopes. Light microscopes use light and lenses to magnify an object. This is the type of microscope you'll typically use in your school lab. Here are the key features you need to remember. The image produced is always in color, and you can often see through the cell, which helps you to observe the internal structures. Light microscopes can be used to observe specimens that are either living or dead, so you can watch living cells in action. They have low resolution, around 200 nanometers, and a low magnifying power, up to around 1,500 times. This means you cannot see structures and organelles in detail like the cell membrane, or see them at all, such as ribosomes. Finally, light microscopes are relatively cheap to buy and maintain, which is why they're so common in schools. Now let's move on to electron microscopes. Electron microscopes are much more advanced. There are two main types: transmission electron microscopes and scanning electron microscopes. Transmission electron microscopes can see through cells, showing their internal structures in detail, and scanning electron microscopes only show the surface of cells. But for now, let's focus on their general features. Electron microscopes use electron beams to magnify an object. This must happen in a vacuum, a space with no particles. This means they can only be used to observe specimens that are dead. The vacuum and the electron beam would destroy living cells. The image produced is always in black and white, no color here. Electron microscopes have an incredibly high resolution, down to 0.2 nanometers, and a much higher magnifying power, up to 100,000 times or even more. However, they are expensive to buy and maintain, which is why you won't find them in most school labs. Now that we know the features of each type, let's compare them side by side. When it comes to cost, light microscopes are cheap, while electron microscopes are expensive. How do they work? Light microscopes use light to illuminate the specimen, whereas electron microscopes use electron beams. Resolution. Light microscopes have a low resolution, and electron microscopes have a high resolution. Image properties. Light microscopes produce true to color images, unless you add a stain, but electron microscopes only produce black and white images. You have to digitally add color to electron micrographs. Magnifying power. Light microscopes have a low magnifying power, while electron microscopes have a high magnifying power. And finally, the specimen you can observe. With light microscopes, the specimen can be alive, but with electron microscopes, the specimen is dead. Here's a quick way to memorize the key comparison points using the phrase CRIMS. Just remember these 6 categories: cost, how it works, resolution, image properties, magnifying power, and specimen you can observe. Try creating a comparison table or a set of flashcards with these headings, and this can help you recall facts for direct comparison of the properties of each type of microscope. Now let's apply what we've learnt with some guided practice. The question reads, explain why an electron microscope is more useful than a light microscope to study the structures inside a cell. This is worth 3 marks. Here's a model answer. An electron microscope has a higher resolution, so you can see more organelles inside the cell compared to a light microscope. Notice how they've clearly stated an advantage, a higher resolution, and linked this to studying structures inside the cell, seeing more organelles. This answer gains 2 marks, but to get 3 out of 3 marks, they should add one more advantage of using an electron microscope. Adding a statement about electron microscopes having a higher magnifying power than light microscopes will score full marks. Now it's time for you to have a go yourself. The question reads, a researcher is studying the structures of a prokaryotic cell. They use a scanning electron microscope, or SEM, which can observe the surface of cells. Discuss the limitations and advantages of using a scanning electron microscope instead of a light microscope for observing the prokaryotic cell. This question is worth 4 marks. When you're ready, pause the video and attempt the question. Think about both advantages and limitations. Press play when you're ready to see the answer. Right, let's go through the answer. To gain 4 marks, aim to state 2 advantages and 2 disadvantages. For the advantages, first, a scanning electron microscope will produce high resolution images. Second, a scanning electron microscope will have a higher magnification power than a light microscope. For the limitations, a scanning electron microscope will produce black and white images, unlike the light microscope, which produces true to color images. Secondly, the prokaryotic cells will be dead with an SEM. You can't observe the living cells. A third mark is for stating that a scanning electron microscope can only show the surface of cells, not the internal structures. Notice how this question asks you to discuss, so you need to give both sides, advantages and limitations. Always read the question carefully. How many marks did you get? Right, well done for completing this video. Today you've successfully learnt how to describe the features of a light and electron microscope and compare the features of light and electron microscopes. If you found this video helpful, please share and give the video a thumbs up and subscribe to stay updated with the rest of the microscopes and magnification series. See you next time.